Welcome to the lecture on fertilization and development. Before we get started, let's talk about just a few simple terms. Whenever we talk about something in science or in this class, we talk about specifically what the appropriate terms are for everything that we're speaking about. So in this case, when we're talking about a developing offspring or a conceptus, and that term means that which is conceived or conceptus. When we're talking about a developing offspring, there's specific terms for this conceptus depending upon what stage it's at. So the first part of a conceptus is, I suppose, a zygote, which is a fertilized egg. And then this begins to develop and becomes an embryo. It starts to divide, and we're going to talk about this in all of the stages of division. This continues to be an embryo until it's about 9 weeks old. And about 9 weeks is the point where we can tell that this developing conceptus is a human. And it's there at 9 weeks that we start calling this organism a fetus. Now we call it a fetus until it's born, and then we either call it an infant or a baby. So I'll try very hard during this lecture to make sure that I use the appropriate terms, either zygote, embryo, fetus, or infant, depending on which stage of development we're talking about. Now, the first thing we have to talk about is fertilization. This is an incredible feat, especially for the sperm, if you think about how far they need to go. Now, they go about 5 inches, 4 inches. Remember, we talked about the length of the vagina in the last reproductive system lecture that I posted. Now, the oocyte, once it's ejected, it's viable for about 12 to 24 hours. The chance of pregnancy drops to almost 0 by the next day. So sperm are actually amazing in that they can survive in the female reproductive tract for up to a week, but fertilization is most likely to occur within 24 to 48 hours after ejaculation. So this means, if you're adding it all up in your mind, that for successful fertilization to happen, intercourse or copulation has to occur no more than 2 days before ovulation and no more than 24 hours after, at which point that oocyte has traveled about 1/3 of the way down the fallopian tube. Remember, we talked about this particular spot is the ampulla of the fallopian or uterine tube. Now, fertilization occurs when the sperm's chromosomes combine with those of an egg to form a fertilized egg or a zygote, like I just mentioned. And this is the first cell of the new organism or individual that's formed. Now, during ejaculation, we've already talked about just how many sperm are in a male's ejaculate. We're talking millions and millions. Despite these vast numbers, most sperm don't actually reach the oocyte, and there's a lot of reasons for this. If you think about everything we've discussed this semester, especially in the immune system and the reproductive system, there's a lot of things to consider. Now, the first is the acidic environment of the vagina. This is necessary for the health of the vagina, which we talked about in our last reproductive system lecture. So, I will say that many of the sperm, frankly, just leak out of the vagina almost immediately. Some of them then go up a woman's urethra, which is often why if a woman's recently had sex, if she gives a urine sample, we will find sperm in her urine. Now, many fall out almost immediately. Several more, thousands and thousands, are destroyed by the vagina's acidic environment. And then others will get stopped because the cervix isn't open. Remember how we discussed how most of the times of the month the cervix has a mucus plug in the os. Now, sperm that do reach the uterus, considering that they make their way through the acidic environment, through the cervix that happens to be open and lacking a mucus plug, that don't fall out, if you think about it, they have to then reside and make their way up the uterus, which is filled with different forms of cells that are designed to destroy them. Remember, the woman's immune system has natural killer cells and neutrophils and all sorts of things that are going, especially phagocytes, that are going to try and destroy the sperm. And thousands more are destroyed by these residents, these parts of a woman's immune system. So only a few thousand sperm out of the millions in male ejaculate are headed their way up the fallopian tubes. But then you have to think half of them go to the wrong one, right? At least half, or maybe less than half, it depends. But Usually women only ovulate into one fallopian or uterine tube. So the other thing to consider is that we do have peristalsis happening by that fallopian tube and cilia movement as well, all trying to move the oocyte down towards the uterus. And so if you think about it, now we have this other additional hurdle in that these sperm, these few thousand that are left, now have to go against the current, so to speak. Now, these difficulties aside, there's other things that sperm need to overcome. And this is that sperm freshly deposited into the vagina and make their way up the fallopian tube, they're not ready yet. They have to undergo this process called capacitation. Now, capacitation involves a lot of different things. Their motility gets enhanced, but one of the most important parts of this is that the membrane surrounding the acrosome in the head of the sperm, this membrane has to become fragile. Now, it becomes fragile once, hopefully, it works its way into the woman's fallopian tube, and it's not fragile before that. So as sperm are moving through the cervical mucus in the uterus and the fallopian tubes in the vagina and all of these things, they actually need the protective membrane proteins, as well as the cholesterol that keeps their acrosomal membranes tough and stable. We need the sperm to be able to make it through and not have those digestive enzymes rupture prematurely in either the male's reproductive tract or somewhere else in the female's. So the sperm that find their way to the oocyte, they actually have to undergo some changes once they get there. And this capacitation, like I said, takes somewhere between 6 to 8 hours. Now, the other question you might be asking is, how do sperm find their way to the egg? And I remember when I was in college, it was sort of still a mystery. And I remember seeing photos like this one on the right where you can see an egg completely surrounded by sperm. And it kind of looks like they know what they found. And the truth is, they do. Sperm have special receptors that sense the direction of fluid flow, temperature, and certain chemicals. These things guide the sperm upstream against the current through hardships unnumbered through the uterine tubes. So, for example, they swim towards warmer temperatures at the far end of the tube, sort of like a dog following a scent trail. And sperm have olfactory receptor proteins that sense or smell chemicals that are released by the oocyte and the surrounding cells in that corona radiata. Another thing I wanted to mention before we talk about actual fertilization is the difference between the sperm that are carrying an X and the sperm that are carrying a Y as far as the sex chromosomes. Now, if you think about it, because the X chromosome is bigger than the Y, the X sperm have slightly more total DNA than the Y sperm, about 2.8% more. And there is a way to take this, as well as the difference in the size of a sperm that have an X versus a Y chromosome. We can use that, and actually many different companies can sort sperm just based on size and shape. And this is how it's used often for any sort of artificial insemination, to sort the X sperm from the Y sperm. So I'm going to draw just a rough picture, and I'll tell you, this is kind of an over-exaggeration, but Y sperm are smaller and faster. Remember, we said that they carry a little bit less DNA. So I'm trying to draw this sperm here on the bottom to look like sort of a sleek Lamborghini, Ferrari sort of race car. This is smaller and faster, but it does die quicker. They live fast and die young, so to speak. An X sperm has sort of the same kind of tail, but the head is much bigger and it contains more cytoplasm. So here's the Y and here's the X. The X sperm are more resilient. They're larger and they're slower, but they have more cytoplasm. That makes them live a bit longer as compared to the Y sperm that live fast and die young. So if you think about it, it's easy to sort X from Y because one's much bigger than the other. All you really need to do is have some sort of mesh or sieve that sorts them, allows the smaller ones to get through, and the bigger ones to not get through the holes. Another way is just to spin them down using a very, very gentle centrifuge, and the weight of them lets them sort out appropriately. Now, there was for a long time this method called the Shuttles method, based after the doctor that figured this out, where people thought that this was sort of a tortoise and the hare situation, where if an egg were a certain spot in the fallopian tube, then the Y sperm could get there faster, and if they were further away, then the Y sperm would die. They wouldn't get there as far, but the X sperm would sort of take their time and eventually get all the way to the end. Now, this isn't very effective because it's very, very hard to time ovulation, and especially based on the variances between individual women. But it's an interesting idea, and it's kind of cool to think about the difference between Y sperm and X sperm and just how easy it is to sort them. Now, when we talk about sperm penetration and fertilization, there's a lot of steps that happen. Remember, we already mentioned that the egg itself, the oocyte, is surrounded by the corona radiata. These are the cells, the support cells that went with that secondary oocyte when it was ovulated. Now, there's also something called a jelly coat or a zona pellucida, which is deep to the corona radiata, and this is just a big layer of glycoproteins in an extracellular matrix. And the contents of the acrosome of a sperm, once it hits the egg, is designed to digest not only some of those corona radiata cells, but also to digest the jelly coat. Okay, so what happens when the sperm reach the egg? Now, a lot of people think that what happens is that one sperm reaches one egg, that's the one that gets in, but that's not really the case. One explosion or one acrosomal discharge with those digestive enzymes is not enough to get through the jelly coat. And the actual process of getting through the corona radiata and the jelly coat has to be the contents of the acrosomal processes of hundreds of sperm. So it's not the first sperm gets the egg, it's sort of the one that was in the right place at the right time, and at least 100 before it paved the way and made a tunnel, so to speak, for that last one to hit the surface of the egg. All right, so once the path has been cleared for a single sperm, the acrosomal process attaches to receptors on the egg. And the acrosomal process for this lucky sperm is covered with a protein called bindin. When a sperm binds to the oocyte's sperm binding membrane receptors, it triggers this thing called the oocyte membrane block. What this means is the oocyte sheds the remaining sperm binding receptors on vesicles made from its plasma membrane. All of these receptors are released. And in the absence of these receptors, additional sperm are unable to bind to the oocyte, even if they happen to make it to the surface. The released vesicles, sort of as a side note, studded with these sperm binding receptors, they almost act as decoys away from the egg that attract and bind any other sperm that might make it through the zona pellucida or the jelly coat. So once the plasma membrane of the sperm and oocyte have fused, there are then waves of calcium that are released from the oocyte's endoplasmic reticulum. It releases this into the cytoplasm. And these calcium surges trigger a cortical reaction, which is the exocytosis of lots of granules located just inside the plasma membrane. These granules contain enzymes that destroy the jelly coat or the zona pellucida's sperm binding receptors, and the calcium also hardens the zona pellucida. So these changes in the zona pellucida, making it really hard like a shell, is called the zona reaction, and it's also called the block to polyspermy. Now, this is a great name for it. This block to polyspermy means that once you have this hardened shell with no receptors for another sperm to bind, this means only one sperm and one sperm only, the one in the right place at the right time, the first one, actually gets in and goes on to fertilize the egg. So this hardened membrane is called a fertilization membrane. We now have fertilization, right? So what happens next is that the sperm, its cytoplasmic contents enter the oocyte, and the sperm loses its plasma membrane. The centrosome from the midpiece of a sperm assembles microtubules, and the sperm uses that to propel its DNA towards the nucleus of the oocyte. On the way towards the oocyte's nucleus, its nucleus swells to about 5 times its previous size, and it forms what we call the male pronucleus. So at the same time, the secondary oocyte is activated from that sort of dormant stage. It was stuck in metaphase 2. These calcium surges trigger the oocyte to do several things. So one thing that it does is that we've seen just figured out in the past couple of years is that there's this shower of zinc ions called, we call them zinc sparks, and you can actually see them with the right kind of imagery. The shower of zinc ions bursts from the egg, and this efflux of zinc allows the oocyte to finally complete meiosis 2. So when it completes meiosis 2, it forms the final egg, the final ovum nucleus, and then that second polar body that eventually gets discarded. All right. So once this is finally finished, the ovum nucleus swells and becomes the female pronucleus, and the 2 pronuclei, both male and female, replicate their DNA as they approach one another. As a mitotic spindle develops between them, the pronuclei, their membranes rupture, their chromosomes are released, and that is truly the moment of fertilization. We now have the maternal and paternal chromosomes combining and producing what we now call a zygote. And it is diploid. Remember, that egg and that sperm were haploid. And when they combine, we've got 23 from the egg, 23 from the sperm. We now have 46, a diploid zygote. Now, one of the questions I always get asked is, what happens? What happens if at the exact moment one sperm is getting in, the other one is getting in at the same time? The truth is, this doesn't happen very often, but if it did, that organism would never become a zygote and start to divide. The reason being that it has way too many chromosomes. Having a third too many chromosomes is a good reason to undergo apoptosis. All right, so let's move on and we'll talk about what happens next. Once we have fertilization, we have ourselves a zygote, then division and embryonic development start to occur. All right, so fertilization is followed by a process called cleavage, and all cleavage means is cell division without growth. That means that that individual organism developing conceptus stays the same tiny little size. And so if it's forming more cells, then each cell gets continually smaller with each division. Now, most animals' embryos nourish themselves with yolk or nutrients in the egg, but humans, we don't really have much yolk and certainly not many nutrients, not enough for this developing embryo to survive. Instead, that developing embryo for us, for humans, has to survive on cytoplasmic components. Remember when we talked about oogenesis, I said that every division to form that oocyte is given in every division. Almost all of the cytoplasm is given to that one developing oocyte, and almost none was given to the polar body. The reason for this, at least in part, is that that cytoplasm is the only thing that this developing embryo has to survive on until it reaches the uterus of the woman. Okay, so we have cleavage, these divisions without growth. Cells get smaller with each division. The first 2 divisions are vertical, resulting in 4 cells. Well, first it's a 2 cell, then a 4 cell. The third division is horizontal and produces an 8-celled embryo with 2 tiers of 4 cells each. And eventually, after about 3 days, continued divisions produce a solid ball of cells, which we call a morula. Now, morula means little berry, which I think is cute. It's a great name, little berry. And this morula, it has been moving, at least this embryo has been moving and developing into a morula all the way down the woman's fallopian tube while this division has been occurring. All right, so the transport has been happening this whole time, and after about 3 days, we have a morula, a solid ball of cells. Now, this little barrier, this little mulberry has been formed, right? And it has many, many cells in there. After about 4 or 5 days after fertilization, the embryo has about 100 cells, and at this point, it's reached the uterus. So what did it survive on this entire way? Remember the cytoplasm that was given to it primarily by the developing oocyte during oogenesis. All right, so what happens once it reaches the uterus is it's now called a blastula or a blastocyst. It's a hollow ball of cells, about 100 cells total. It stays in the uterus for a couple of days and continues to develop. But even at this stage, we have this blastula or this blastocyst. It has a lot of different components to it, which we're going to talk about in just a minute. But one component I do want to point out that you can see on this picture is the blastocele, which is the hollow fluid-filled cavity inside of the cells themselves. Now, again, this takes about 4 or 5 days, and it reaches the uterus. And here is where it starts to finally get some nourishment from the uterus itself, from some of the fluids and things inside of the uterus. All right. So, in the uterus, and we can see what a journey it has become. Remember, this developing oocyte was ejected from the ovary, caught by the fimbriae, and started to travel by both peristalsis and movement of cilia down the fallopian or the uterine tube. It does, well, the idea is that it will get fertilized. Remember, it's been arrested in metaphase 2, that it will get fertilized in the way that we've just spoken about in the top third of the fallopian tube. When it does this, the oocyte finishes meiosis and creates that second polar body. We now have a zygote, and this continues to undergo cleavage as it moves down the fallopian tube until it reaches the uterus. Now remember, we have a few stages. After about 3 days, we call it a morula, a solid ball of cells. And when it reaches the uterus after 4 or 5 days, we call this a blastula or a blastocyst. This has a blastocyst cavity or a blastocele, this space inside of it. If we look over here to the right, you can see that what happens next is this blastocyst will eventually implant itself into the lining of the uterus, which, if you remember, is called the endometrium. Now, the blastocyst does stick around for a couple days, 2 or 3 days, and it's nourished by the glycoprotein-rich uterine secretions inside of a woman's uterus. Then about 6 to 7 days after ovulation, finally implantation begins. And there's a lot of things that have to have happened to the lining of the uterus that we talked about last time, right? That are primarily prompted by surging levels of estrogen and progesterone made in the ovaries by those granulosa cells. All right. So if the mucosal lining, the endometrium of the uterine is receptive, cell adhesion molecules bind to this part of this blastocyst that is the outer part here. And we call this outer part the trophoblast. Now this trophoblast is what binds to the cell adhesion molecules on the endometrium. And you can also see, not only do we have this space in here called the blastocele or the blastocyst cavity, we also have the beginning of that developing embryo, what's going to be eventually the fetus and the infant. It's called the inner cell mass, the inner cell mass. So right here, only a week after fertilization, we now have a real delineation. We have the outer part, which is called the trophoblast. We've got this inner cell mass, and we have this fluid-filled space called the blastocyst cavity or the blastocele. Now, the trophoblast cells bind to and adhere to the endometrium, and they secrete digestive enzymes and growth factors onto the surface of the endometrium. The endometrium quickly thickens at this point of contact and almost takes on the characteristics of an inflammatory response. The blood vessels become more permeable and leaky. Inflammatory cells like lymphocytes, natural killer cells, and even macrophages invade the area. The trophoblast then has some changes that occur in it. And it forms a couple of distinct layers, which we're really not going to get into. But I will say that as the endometrium becomes eroded, the blastocyst then has its chance to burrow into its thick, bloody, mucousy, juicy lining and is surrounded by a pool of blood leaked from those degraded endometrial blood vessels. So eventually, the blastula or blastocyst dives in and implants so deep that it's completely embedded and covered by the endometrium. I call it sort of digging in like a tick, but that's because I'm from Iowa. As exciting as this all is, I should probably tell you that at least 2/3 of all zygotes formed fail to implant by the end of the first week or spontaneously abort. If that wasn't enough, about 30% of implanted embryos later miscarry due to some sort of genetic defect of the embryo or some sort of problem with the uterus itself. A number of different things can happen. So it really is incredible. Not only what had to occur for an oocyte to get released from an ovary and for the sperm to make it all the way to the top third of the fallopian tube, but then also how many don't make it even if all of that happens and it makes its way after all of the divisions to the uterus itself. Now, I just to repeat myself, about at least 2/3 of all zygotes formed don't implant by the end of the first week and spontaneously abort. And then about 30% of those that do implant later miscarry due to problems either with the embryo or the uterus or something else. So, successful implantation takes about 5 days, and it's usually completed around day 12, which, if you're keeping track, is day 26 of a woman's menstrual cycle. And this would be just before the endometrium normally would slough. So this would be bad. Menstruation, if that occurred, would flush away that newly implanted embryo. So the embryo itself takes special measures to make sure that the corpus luteum doesn't go away. Now, don't forget the corpus luteum that's left behind from that released oocyte. This is creating lots of progesterone and estrogen, which maintains the uterine lining. So what does this? It is the embryo itself. It secretes a hormone called human chorionic gonadotropin, or HCG. Human chorionic gonadotropin acts a lot like luteinizing hormone, but doesn't have to go through the hypothalamus, pituitary, ovary controls. What it does is it keeps the corpus luteum alive and making progesterone and estrogen, which is incredibly important to maintain that thick, juicy, bloody uterine lining that this embryo has just implanted into. This HCG, or human chorionic gonadotropin, is what pregnancy tests generally are based upon. They detect this hormone in a woman's blood or urine. Now, this HCG is usually detectable in a mother's blood around 2 days after implantation, which is when pregnancy tests are considered pretty accurate. Blood levels of HCG continue to rise until about the end of the second month, and then the blood levels decline sharply once the placenta is established, which we'll talk about later. But the placenta assumes the role of making progesterone and estrogen, so there's no need to keep that corpus luteum going. It's allowed to degenerate, and the ovaries still remain inactive until after birth because of all of the progesterone and estrogen being made by the placenta. All right, so implantation, like I said, occurs 6 to 7 days after ovulation. Just a quick reminder of some of our terms here. We have the trophoblast, which is the outer ring of cells, and this trophoblast will eventually become the chorion, which is the embryonic or fetal part of the placenta. We have the inner cell mass, which is going to be that developing little organism. And we have the blastocele or the blastocyst cavity, that fluid-filled space inside of the blastocyst. Now, the cartoon version is interesting, but look at the real deal over here on the right, and then look how it implants and embeds itself into the woman's endometrium. Again, it takes about 5 days, and it's completed by about 12 days after ovulation. Now, while implantation is occurring, there's lots of things still happening. This embryo is still developing, and we call the next stage of embryonic development gastrulation. So what happens here, if we look down below, here we have our blastula or our blastocele, which doesn't look 100% accurate anymore. A gastrula is just a blastula, but a little bit more developed. And if you look down here, you can see that a blastula becomes a gastrula as the cells begin to invaginate or buckle inwards. This thing that buckles inwards, we call the archenteron. Archenteron. What a great word, right? You can see that right here, archenteron. So as it invaginates or buckles inwards, it creates a hole or an opening at one end, which we call the blastopore. And this eventually becomes the anus in the organism. Not trying to make you feel bad about yourself or anything, but really, well, if we're being honest, the first thing that formed in you was your anus. So the cells rearrange and invaginate further. Remember, this invagination we call the archenteron. And there are 3 distinct cell layers present at the completion of gastrulation. These are called germ layers. These germ layers include the endoderm, mesoderm, and ectoderm. And at this point, each of those germ layers is now predestined to become a specific part of the body. All right. So this one other thing that you can see before we move on, other than the endoderm, mesoderm, and ectoderm, is that there is, if we look up here above, you can see we have this yolk sac that's forming. We have the amnion that's forming, a fluid-filled sac called amniotic fluid. It's forming around the developing embryo. And then this other thing called the allantois, which we're going to talk about here in just a second. All right, before we talk about those structures, I want to point out that those 3 germ layers, endoderm, ectoderm, and mesoderm, really are predestined to become something. And if we look at our late gastrula, once gastrulation is complete, you can see the outer portion of the cells is the ectoderm. The innermost lining is called the endoderm, and the middle layer in pink is the mesoderm. And if you look at this table here, you can see what each one of those layers is predestined to become. Now, if you are in Rachel Willard's class, I'd like you to flip over and look at the mini lecture that somebody has provided to you on the embryonic germ layers. For the rest of you, let me just point out a few key things, like the ectoderm becomes the nervous system, and some of the more outer layers like the epidermis of the skin, hair and nails, and tooth enamel. The mesoderm becomes primarily your muscles and your bones and most of your basic systems. And then the endoderm becomes most of the linings and some of the deep glands like your thyroid and parathyroid glands. So the first organs to be produced, and by this we're now talking about organogenesis, or the formation of new organs. We're also talking about neurulation because the first organs to really form are those of our nervous system. The first organs to be produced in chordates, that is, organisms that have a spinal cord, are the neural tube and the notochord. Gastrulation lays down the basic structural framework of the embryo. It also sets the stage for rearrangements that occur during this process of organogenesis. I will say that by the end of the embryonic period, at about 8 weeks, when the embryo is only slightly more than just an inch long from head to butt, that's referred to as the crown to rump measurement, if you can believe it. All of the adult organ systems are recognizable, and there are actual brain waves that have been found and recorded from this 8-week embryo. So the first thing that happens during organogenesis and neurulation is that the embryo starts off as a flat plate. And as it grows, it folds to form sort of a cylindrical shape. This cylindrical shape, which lifts off of the yolk sac, it protrudes into that amniotic cavity, that fluid-filled space. So this sort of resembles, if you think about the three layers here, endoderm, ectoderm, and mesoderm, sort of like three stacked sheets of paper that begin to fold laterally into a tube. At the same time, folding occurs from both ends, the head and the tail regions, and progresses towards the central part of that embryonic body. There's where the yolk sac and umbilical vessels will eventually protrude. Now, what happens is that the endoderm eventually undercuts, and as its edges come together and fuse, it encloses part of the yolk sac. Now, before we get to that, let's talk about this formation of the neural tube and the notochord. Now, the first major event in organogenesis is neurulation, and this basically comes from the ectoderm, and the ectoderm produces the brain and spinal cord. This process is induced or stimulated to happen by chemical signals from the notochord, which is a rod of mesoderm that defines the body's axis. The ectoderm on top of the notochord, here we can see the little notochord, it thickens and forms the neural plate. And then the ectoderm folds, forming a groove, which we call the neural groove, until it eventually folds and those folds fuse on themselves, forming a tube, which we call the neural tube. And this tube soon pinches off from the ectodermal layer and becomes covered by surface ectoderm. The anterior end of the neural tube becomes your brain, and the rest becomes spinal cord. The associated neural crest cells with it migrate and give rise to cranial, spinal nerves, other sympathetic ganglia, cells in the adrenal medulla, pigment cells in your skin, all sorts of things. But it really is the combination of the notochord and the plate of dorsal ectoderm rolling itself into this tube that becomes your brain and spinal cord. The midline cells on either side here, let me erase a little bit. On either side of the notochord, these cells are called midline endodermal cells, these cells here on the side of the notochord. These are called somites and eventually become segmental muscles and vertebrae. And if you can believe it, this process is similar in all chordates, all organisms with a spinal cord, which is why I have a picture of our little friend, the frog. Now, this is a really complicated process, and as complicated as it probably just sounded, it is much, much more complicated than that. So I would recommend that you take a look at your book and just read through these processes again. Now, we just talked about how we start to begin the process of organogenesis and neurulation, the formation of our brain and spinal cord, and some of our vertebrae and back muscles, how they're the first things to really form in a primitive way. We also have several extra embryonic membranes that form the first 2 to 3 weeks of development. These are happening at the same time that we're forming our organs and our neural structures. So I already mentioned that the amnion forms, which is what's colored in all of these images in sort of the baby blue color. And this amnion is a transparent membranous sac that becomes filled with a really slick sort of cirrus-like fluid called amniotic fluid. And it surrounds the entire embryo, providing cushioning and lubrication for developing little parts like fingers and toes and eyelids. It helps with temperature regulation, and especially later in development, allows a developing fetus a place to urinate into. This is eventually going to become what the water is when a woman's water breaks, when she gives birth. This sac is almost always broken before a baby can be born. And that fluid that comes out, that amniotic fluid, we call water breaking. But we know now that it's not really just water. It's a lot of different fluids that has proteins, and it's slick proteins, but also a good amount of fetus pee in there as well. Now we also have the yolk sac that you can see is in yellow. This yolk sac hangs off the ventral surface of the embryo and has very little yolk in humans. But we use it anyway. What we do is we pull it inside of ourselves and use that to become part of our digestive system or our gut. We also use it to help produce blood cells and create some of our blood vessels, especially associated with our digestive system. You can also see, hanging off the ventral surface of that developing embryo, you can see this sort of stem-like structure, and this is an extension of embryonic tissue at one end of the yolk sac, and this becomes the umbilical cord, which will contain the umbilical artery and vein. We have already talked about how the trophoblast, that outer ring of cells on a blastula, eventually becomes the chorion. And the chorion is what is the fetal or embryonic part of the placenta. So that is, you can see in this picture, as sort of these outcroppings all around that developing embryo that insert itself into the endometrium and contain lots of blood vessels. Now let's talk about the process of creating a placenta. I will say that this is one of the most incredible things, is that a woman can literally create this unbelievable organ that is an endocrine organ and a cardiovascular organ, and in most cases almost the size of a dinner plate, can create this in 12 weeks or less. So a placenta is a combination of both embryonic and maternal tissues. Now the chorion, which remember came from the trophoblast, it develops these little finger-like projections. And if you look in the top right, you can see that we have these little projections that are essentially outcroppings or outgrowths of that trophoblast, of that chorion, that project into the endometrium of a woman. All right, we call these chorionic villi. Remember, villus means finger-like. And these villi project towards the embryo, or they project towards the endometrium. Some project towards the embryo, and we call that the allantois, which we've already mentioned, and this eventually becomes the umbilical cord. Now, in between these chorionic villi are the spaces of the woman's or the mother's endometrium, and we call these intervillous spaces. Remember, these are endometrium, and they're in the stratum functionalis. The villi of the developing embryo of the chorion lie in these spaces, immersed in maternal blood. So the part of the endometrium in between the chorionic villi we call the decidua basalis. And if we really are being accurate, it's the decidua basalis plus the chorionic villi that make up the placenta. So the placenta essentially is half maternal and half embryonic, with the maternal part of it being the intervillous spaces or the decidua basalis, and then the chorionic villi, those outcroppings of the chorion, those are the embryonic parts of the placenta, and it's the two of them together that are the placenta themselves. So that's placentation, or the production or formation of a placenta. And like I said, this takes somewhere around 12 weeks. Now, just now that we've been through all of this, let's do a little bit of a review on what has happened through the weeks. We've made it quite a ways. Remember, we said that all of the organs are in place and neurulation has happened, a placenta is formed. So let's just do a little bit of a review here. So week 1, what happens? Fertilization happens in the upper third of the oviduct, the fallopian tube, the uterine tube. Remember, we have to have 3 names for everything in anatomy. This results in cleavage, and the embryo passes down the fallopian tube until eventually it reaches the uterus. Remember, it's a morula when it reaches the uterus on the 3rd day. It's a blastula after 5 days, and then it starts to implant. Here's what it looks like after about 4 days. And I love this picture because it shows you that, remember, 3 days is a morula and 5 days is a blastula. What do you call this? And the answer is I'm sure somebody has a million names for it. I call it in-betweeny. It's in between a morula and a blastula. And I show this picture just so that you see it's not like Shazam. It goes from a hollow ball of, or from a solid ball of cells to a hollow ball of cells. It does take lots and lots of divisions and organization. By week 2, 1 week after fertilization, the blastocyst implants in the endometrium. Gastrulation occurs. Don't forget that HCG is secreted from that developing embryo to keep the corpus luteum intact. Estrogen and progesterone continuously released by that corpus luteum, and that keeps the uterine lining thick and juicy and bloody and mucousy for that implanting embryo. At this point, the amniotic cavity and the yolk sac form. And here, I guess, is a cartoon version of what that looks like. It doesn't look like much, but if you think about it, boy, it's a lot just in 2 weeks. We've got the trophoblast, the outer part that will eventually become the chorion, or the embryonic part of a placenta, the fetal part of a placenta. We've got the yolk sac next to the embryonic disc, which eventually will become that fetus. We've got the amniotic cavity filled with fluid for cushioning and temperature regulation. We have the blastocele, the cavity there. And by week 3, remember we said that neurulation occurs. We also have heart and blood vessels beginning to develop. You can see on this picture here, the real deal. You can see the neural groove. Let me see if I can find a different color here. Let's go with blue. You can see the neural groove on either end, the anterior and the posterior end, and eventually this groove will roll into a tube. You can see from our picture down below what's happening here. You can see that the chorionic villi are projecting into the endometrium, into the decidua basalis. You can see that the yolk sac is now slightly started to be pulled in and will be used to make parts of the gut and create blood cells. You can see that developing little embryo there surrounded by fluid in the amniotic cavity. And you can even see the allantosis here now that eventually will become the umbilical cord. So, as long as we're talking about the cardiovascular system, we can talk about some of the special things that are present in a fetus and in fetal circulation. Embryonic development of the cardiovascular system lays the groundwork for the fetal circulatory system, which is converted to the adult pattern at birth. A lot of changes. Now, remember I just said the first blood cells really do arise in the yolk sac. We make good use of that. And before week 3 of development, tiny spaces appear in the mesoderm. These are quickly lined by endothelial cells, and eventually these become vascular networks that are destined to become the heart, the blood vessels, and your lymphatic vessels. By the end of week 3, the embryo has a system of paired blood vessels, and the 2 vessels forming the heart have fused. By 22 days of development, a miniature heart is pumping blood for an embryo less than 2 millimeters long, if you can believe it. Now, there's a lot of unique things about fetal circulation or prenatal development, and that includes the umbilical artery and veins, and then 3 vascular shunts that we're going to talk about. Now, the umbilical arteries and veins are a little different, just like when we talked about the pulmonary arteries and veins. They don't necessarily carry what you would expect. Umbilical arteries carry deoxygenated blood from the fetus to the placenta, and umbilical veins carry oxygenated blood from placenta to fetus. So again, this is a little bit opposite of what you might be thinking. These are in what once was the allantosis and eventually becomes the umbilical cord. Now, we also have some other shunts. The ductus arteriosus transfers most of the blood from the pulmonary trunk directly to the aorta, which allows blood to bypass the lungs. The lungs are one of the last things to develop in a developing fetus, so it makes sense that there would be no need to or any reason to bring blood to the lungs because they're not functioning. All of the gas exchange has to happen in the mother's lungs. The foramen ovale is another shunt, and this is a hole in between the atria. Blood entering the right atrium flows directly into the left atrium, and this also allows blood to bypass the lungs because they aren't working and done yet. And then the last shunt is the ductus venosus. Blood from the umbilical vein, most of it enters this, and this allows blood to bypass the liver because the liver is another structure that just doesn't finish until very late in development. This means that in a fetus, the inferior vena cava contains mixed blood from the right atrium. So this mixed blood goes directly into the right atrium, and what I want to talk about now is what happens when that infant is born. So these shunts and these umbilical arteries and vein, they aren't needed anymore. And so a lot of things have to happen. At birth, the umbilical blood vessels, fetal shunts are no longer necessary. And the umbilical arteries and veins constrict and become fibrosed within a minute or so of that baby taking its first breath. We also have the umbilical vein in part becomes the round ligament of the liver, which you guys have all seen on the models. This helped to attach the umbilicus to the liver. And if you ever have the chance to dissect a cadaver or another organism, you'll often see that this round ligament is attached to the liver. The ductus venosus collapses as blood stops flowing through the umbilical vein, and eventually it's converted into ligamentum venosum on the liver's undersurface. All right, as the pulmonary circulation becomes functional in a newborn infant, pressure on the left side of the heart increases, the right side decreases a little bit, and this causes the pulmonary shunts to close. The flap of the foramen ovale is pushed to the shut position, and the edges fuse to the wall of the atrial septum. It usually results in just a slight depression where we now call that the fossa ovalis. That's what used to be the fossa ovale. And obviously there are times when this doesn't completely close, and often surgery is necessary to close it if it doesn't resolve on its own quickly. The ductus arteriosus constricts and eventually is converted to a cord-like ligamentum arteriosum that just connects the aorta and pulmonary trunk, but no blood flows in between the two. All right, so that's a lot of changes that have to happen within minutes, no more than 30 minutes after birth. Closure of the foramen ovale, it happens slowly, but usually there's no real blood flow transferred between the left and right atrium. By the end of the embryonic period, about 8 weeks, all of the bones have begun to ossify and the skeletal muscles are well formed and they're contracting. Kidneys are developed, gonads are developed, lungs and digestive organs are attaining their final shape. Blood delivery is happening in between the placenta and the mom and the developing fetus via the umbilical vessels. Heart and liver are forming. All of this is by the end of 8 weeks in an embryo, slightly more than 1 inch in size from crown to rump. Now, let's back up just a little bit and talk about those weeks 4 and 5. I think that this is really interesting to take a look at just what we look like between weeks 4 and 5. Remember, we're not called a fetus yet. We're still an embryo. It's not quite apparent that we're human at this point. We have that umbilical cord you can see here. Here's the umbilical cord. You can see that we have tiny little flippers called limb buds. These are our arms and legs developing, our little limb buds. You can see we have quite a large head with a brain in there. We have optic vesicles, which are our eyes developing. You can see we have the liver and the heart developing, blood vessels as well. You can see we have a glorious tail that we form, which eventually undergoes apoptosis. At least most of it does, at least in humans. Of course, in other animals, many of them maintain that tail. We also have a pharyngeal pouch, which in some organisms goes on to become gills, but in us becomes some of our glands, like our thyroid gland. And here's a pretty picture of a 5-week embryo. Now, I don't know about you, but I can see a lot of things here. I can see a nose forming. I can see a mandible. I can see the optic vesicle where the eyes are. I can see blood vessels in there. I can see a brain forming. And if we look over here on the right, you can see sort of the cartoony version again of what's occurring, where we have the yolk sac being pulled almost entirely inside. You can see the developing embryo, the little blue thing. You can see the chorionic villi extending into the decidua basalis, the amniotic fluid surrounding the embryo. Really, a lot has happened in 5 weeks. Weeks 6 through 8, brain and neck development, the nervous systems develop, the organ systems are established, and you can definitely tell it's human. The embryo is now a fetus by the time we hit 9 weeks. And if you take a look at this, how pretty is that amniotic sac filled with fluid? You can definitely tell this is human. There's little toes and fingers, and you can see there's the eyes forming. You can see the umbilical cord, and here is our placenta on the left. Now, the placenta itself is finished by week 10, and don't forget this is how material diffuses between maternal and fetal circulation. It provides nutrients, gets rid of waste, gas transfer for that embryo. Unfortunately, harmful chemicals can pass through the placenta as well, things like nicotine and toxins and all sorts of things, which makes it incredibly important for a woman to watch what she eats and drinks. The placenta produces progesterone and estrogen, incredible high amounts, so it's no longer necessary for HCG to be released by the embryo, and the corpus luteum eventually degrades. Now, these high levels of progesterone and estrogen not only keep the uterine lining nice and thick and juicy, but it prevents new follicles from forming, so that means a woman can't get pregnant while she's pregnant. If you look over here on the right, you can see we have this developing fetus, and here's the placenta. Now, the placenta can be anywhere on the uterus, and there are certainly places that are more desirable than others. Let's just talk about places that are undesirable. If you can imagine, one of the worst places for a placenta to be would be over the cervix itself. There's also situations where during delivery, based on where the placenta is located, it could cause possible problems like the umbilical cord gets wrapped around the fetus somewhere and can cause distress. In those cases, a woman may need to have a C-section or something like that. So months 3 through 9, there is just way too much to list, you guys. Continued fetal development, genitalia appear in about the 3rd month, and a fetus that's 6 months old can survive on a respirator. The lungs aren't fully developed yet, nor the liver at this point. There's a lot of weight gain during the last 2 months for the woman, but also the developing fetus. And in fact, one of the biggest cravings, and that's why I have this donut here, one of the biggest cravings for a woman in her last 2 months of pregnancy is for fat. And this is when that developing fetus's nervous system, all of the axons are myelinating. And if you remember, myelin has lipids in it. So the desire for fat is incredibly high, and the demand for fat because of this myelination is incredibly high. I've actually read in many places that a woman could not be pregnant past 9 months because she couldn't eat enough to keep up with the demands of that developing fetus. All right, now I don't know why this is called 4D. Maybe one of you can explain it, but certainly we can use lots of different types of technology to check in on that developing fetus. We use things like ultrasounds. This is what's called a 4D ultrasound. I don't know what the 4th dimension is, but it certainly is cute if you look over here at this one yawning. But it's a great way to diagnose certain developmental and physical problems. We also have things called amniocentesis, where we can go in with a needle through the abdomen of the pregnant mom and take some fluid from the amniotic sac, which contains cells from that developing fetus, and then analyze those cells for genetic defects. It's not done quite as often because we have incredible blood tests that we can do, and maternal circulation often contains a certain amount of the fetal blood cells. So sometimes that's enough that we can gather genetic information just from drawing blood from mom. Now, if we take a look at the hormones present when a woman is pregnant, if we just look here, I want you to look at the orange, which is HCG, or human chorionic gonadotropin. Remember, this is made by the embryo itself, and this peaks at about 6 to 8 weeks. And this, of course, is to keep the corpus luteum intact and creating estrogen and progesterone. By the time we hit, between 10 and 12 weeks, this amount goes down drastically. And that's because the placenta is now established, and it being an incredible endocrine organ as well as cardiovascular, creates unbelievably high amounts of estrogen and progesterone both, which not only keep the uterine lining thick and rich and juicy, but also prevents a woman from being able to get pregnant while she's pregnant. She will not ovulate when she makes estrogen and progesterone at this high of a level. You can see when a woman gives birth, because she not only gives birth to the fetus, she also gives birth to the placenta, that these hormone levels all go down to a very low level. And it's believed that these tanking levels of hormones are what contributes to postpartum depression. The mean duration of pregnancy is about 38 weeks from the time of ovulation to birth. Pregnancy, it involves a lot of profound adaptations that occur in almost every single body system. Not only are there anatomical changes, but there's changes in metabolism and physiology. New organs are grown, not only to support the pregnancy and the developing embryo and fetus, but also to prepare her body for delivery and also lactation. So as pregnancy progresses, female reproductive organs become more vascularized and engorged with blood. The vagina develops a purplish hue and her vulva a purplish hue, which is called Chadwick's sign. The breasts enlarge and engorge with blood, and their areolae darken. This is because of the rising levels of estrogen and progesterone. Some women develop increased pigmentation of their facial skin on their nose and cheeks, a condition called chloasma, or the mask of pregnancy. There's other things that occur. The abdominal organs obviously have to shift. If you take a look at this picture here, you can see how unbelievable the uterus expands with every consecutive month. So organs definitely have to shift, with the stomach being pressed up against the diaphragm and the uterus eventually sitting and weighing on the bladder. This can create a lot of discomfort. Women have a hard time eating very large meals. Often they have a lot of pressure on their bladder and they have to urinate a lot, or they get stress incontinence. A hormone called relaxin gets released by the placenta, especially further into the pregnancy. This causes pelvic ligaments and the pubic symphysis to relax, widen, and become more flexible, because if you think about it, the actual pelvis dislocates, and it would tear a woman apart if these ligaments and pubic symphysis didn't relax and become flexible. A woman gains a significant amount of weight. This is due not only to the developing embryo and fetus, but also the fluids and the placenta and the uterus itself all growing larger and growing. Often this weight gain and the shifting of abdominal contents can cause lordosis, which is the spinal column being pulled more anteriorly. This is especially apparent in the 7th, 8th, and 9th months of pregnancy, and you will often see pregnant women holding their lower back because of the pain from the lordosis. There's also a couple other hormones secreted from the placenta called HPL and HCS, human placental lactogen and human chorionic somatomammatropin. These not only promote the growth of the fetus, they stimulate the breast to mature further, and it causes the mother's maternal cells to metabolize more fatty acids and less glucose, giving the rich, wonderful glucose to the fetus itself. Other systems that are affected by pregnancy, I mean, we could say all of them, but certainly there is morning sickness, especially in the first trimester. A lot of doctors say that that's due to getting used to some of those pregnancy hormones we've mentioned. Difficulty breathing because of the increased task on the respiratory system of the mother, having to breathe and do gas exchange for two. Same goes with the cardiovascular system. The skeletal system suffers. Not only the spinal column, but the arches of the feet can often fall because of the weight and the stress on them. Stretch marks, especially on the stomach, from the skin growing so quickly. Let's see. I'm sure all of you that have had a baby or been around somebody who was pregnant could definitely give me some more information. Constipation, heartburn, all sorts of things. Many women have a heightened sense of smell that many think is designed just to prevent her from consuming anything that could harm the developing embryo or fetus. Here's just another image from your textbook showing what the uterus looks like before conception. And then, of course, at 9 months, where we have the fundus of the uterus butting up against the xiphoid process, people, the xiphoid process. Now, parturition or birth is a complicated process. And if you're in Rachel Willard's class, please turn over to the mini lecture on parturition, which is the fancy name for birth or delivery. So a fetus largely determines its own birth date, and several events and hormones interlock to trigger labor. It's a combination of hormones released from the fetal hypothalamus and adrenal glands as they mature during the last few weeks of pregnancy, as well as some hormones released by the placenta that create a situation where we have a rapid increase in estrogen production by the placenta. Now, the placenta produces both estrogens and progesterone, and these levels are constantly increasing through the pregnancy. A major function of progesterone during pregnancy was to suppress contraction of the uterine smooth muscle as it's stretched for the growing fetus. But as pregnancy continues, the quieting action of progesterone is important for preventing preterm labor. And as estrogen levels rise at the end of pregnancy, they switch the progesterone receptors to a less effective type. The increase in estrogen has a lot of effects, including preparing the cervix for dilation. It prepares the myometrium of the uterus for contraction. Remember the muscular layer. And it increases the sensitivity of the myometrium to chemical messengers that will cause it to contract. Primarily one of these being oxytocin, which you know is one of, and prostaglandins, which are the two main things that are chemical messengers that are going to drive the muscle in the uterus to contract during labor. Now, as birth nears, these uterine muscles that have been growing and are sensitive to prostaglandins and oxytocin, under the influence of estrogen, the placenta releases prostaglandins that start to trigger the rhythmic expulsive contractions of true labor. Eventually, pressure from the baby's head dilates the cervix, and this increases the cervical distension, and it activates the mother's hypothalamus, which then signals for oxytocin release by the mom's posterior pituitary gland. Now, remember, labor is a positive feedback mechanism, and this involves both prostaglandins and oxytocin constantly propelled into action. And so each contraction creates further muscle contractions, and each distension of the cervix causes a release of more prostaglandins and oxytocin, which creates more muscle contraction and more cervical dilation. And well, you get the idea. So prostaglandins and oxytocin, as well as estrogen, are all responsible for labor. Now, these increased estrogen levels and these oxytocin receptors are what cause Braxton Hicks contractions. They're just this irritability in the uterus itself. But eventually, when true labor happens, the cervix dilates and the amniotic sac ruptures. Labor includes dilation, expulsion, and placental stages. Now, if we look here on the next slide, you can see that these three stages involve first dilation. Now, dilation may not seem like that big a deal, but we're talking about this cervix being fully dilated by the baby's head, about 10 centimeters in diameter. As labor starts, these contractions not only are done by the uterus, but the cervix begins to dilate and open, and the woman loses the mucus plug, which is present through all of pregnancy. When the mucus plug goes, the next thing that usually happens due to the cervical dilation and the muscular contractions is that the amnion ruptures and releases the amniotic fluid, which I mentioned before is commonly called a woman's water breaking. Now, dilation stage is usually the longest part of labor, lasting 6 to 12 hours or more. I know my sister had her dilation stage. It was 4 days long. So certainly lots of events have to happen for this to occur. So engagement occurs when the infant's head enters the true pelvis of a woman, and the descent continues through the vagina. The baby's head rotates generally so that its greatest dimension is in the anterior-posterior line, which allows it to navigate those narrow dimensions of the pelvic outlet of the mom. Now, the expulsion stage lasts from full dilation to delivery of the infant, or actual childbirth. And by the time the cervix is fully dilated, strong contractions are occurring about every 2 to 3 minutes and lasting about a full minute. So in this stage, it can last about 2 hours, it can last an hour, it can last 5 minutes. It depends upon the woman. Crowning's when the largest dimensions of the baby's head distends the vulva, and at this point, sometimes an episiotomy is necessary. This is basically when an incision is made to widen the vaginal orifice, and often this is done in the perineum, or the space in between the vaginal orifice and the rectum. Now, these days, most doctors won't do episiotomies. They've found that healing is a lot faster with a lot less scarring if they let a woman tear naturally. And in the end, a woman may not tear as much as the damage they would do with an episiotomy. An epidural is something that also can occur during labor, and this is when anesthesia is put into the epidural space in the lumbar region of the spinal cord. And this doesn't completely numb the pain, but it certainly cuts it down significantly. So as the baby emerges, sometimes it's not always head first. Sometimes if it's butt or legs first, we call that breech. There's also a condition, especially in cases where the placenta is in a bad position, or there's a breech position of the baby. A cesarean section is necessary, and this is where the infant is removed by surgical removal, cutting through a woman's abdominal wall and her uterus itself. This, interestingly, is named after Julius Caesar, who was, being about to be born, and he was going to be the emperor of Rome. Because they were so concerned about childbirth back then, they actually killed his mother, more concerned for the developing baby. And that was when the first cesarean section was titled or labeled. Now, I don't know how true that is, but it is where the name comes from. It's after Julius Caesar. All right, so crowning occurs when the largest dimension of the baby's head distends the vulva. The baby's neck extends as the head exits from the perineum, and once the head has been delivered, the rest of the baby's body is delivered much more easily. After birth, the umbilical cord is clamped and then cut. All right, so the next thing that has to happen, of course, is that the placenta has to be delivered, and this is attached to all the fetal membranes. This collectively is called the afterbirth. So this usually happens about 30 minutes after the birth of the infant, and there are another series of strong uterine contractions that continue after birth, and these help to further compress uterine blood vessels and limit bleeding. It's incredibly important once the placenta is delivered that every piece of the fetal membranes and the placenta are checked, and there are people usually in the birthing room that do this just to make sure that no piece of the placenta or membranes are left behind in the mother. If this is the case, then the mother could have incredible complications, including infection and sepsis. All right, so just a little reminder that once a baby's born, the umbilical arteries and veins constrict and become fibrosed, the shunts become ligaments, and that foramen ovale closes. At least that's the general idea of what should happen. Just a reminder that one of the most important things that make these umbilical arteries and veins constrict and become fibrosed and the shunts close is that baby taking his or her first breath. This is the crucial first requirement of birth, and this stimulates all of these things to happen. The lungs staying inflated is much more difficult for premature infants because often surfactant production isn't happening yet. We'll talk about preemies here in just a minute. At 1 and 5 minutes after birth, the infant's physical status is assessed and based on 5 main signs: heart rate, respiration, color, muscle tone, and reflexes. And each observation is given a score of 0 to 2. The total is called the APGAR score. An APGAR score of 8 to 10 indicates a healthy baby. Anything lower than that can reveal problems or one of more of their physiological functions assessed and checked out. So about 6 to 8 hours after birth, infants pass through an unstable transition period. Usually they have increased activity, alternated periods of increased activity and sleep. During the activity periods, vital signs are irregular, and often the baby can even gag or throw up a lot of mucus and debris. Eventually, after this, the infant stabilizes a bit, and having waking periods about every 3 to 4 hours, which is generally dictated by hunger and the need to feed. Now, speaking of feeding, the primary way that newborn babies are fed, at least ideally, is lactation. Now, not all women are able to lactate, but certainly that is the hope. Prolactin secretion stimulates lactation after about 2 or 3 days. Rising levels of estrogen, progesterone, and human placental lactogen towards the end of pregnancy, they stimulate the hypothalamus to release prolactin-releasing hormones. The anterior pituitary gland then responds by secreting prolactin. And we talked about this when we discussed the endocrine system. After a delay of 2 or 3 days, like I said, true milk production begins. Before that, and during that initial delay, the mammary glands make this yellowish, brownish fluid called colostrum. This has a lot less lactose than milk and almost no fat, but it contains more protein, vitamin A, and minerals than true milk. Like milk, it has a lot of antibodies in it, and these help an infant's digestive tract and helps them fight bacterial infections. Now, what are some advantages for breastfeeding or with breast milk? And that is just a few things. Its fats and iron are better absorbed, and its amino acids are metabolized more efficiently than those of other animals' milk and formula. Has a lot of beneficial chemicals, including antibodies that are passed through the mother's breast milk. There are lysozymes and interferons and lactoperoxidase, something that protects infants from life-threatening infections. There's also interleukins, prostaglandins. There's bacteria in there, especially beneficial ones that help colonize the infant's gut. It contains a natural laxative that helps cleanse the bowels of this tarry green-black paste called meconium. That was in there before the baby was born. Other advantages: it helps a woman to lose weight. It helps her to stimulate the uterus to go back to its normal size. It certainly is a sweet experience, a bonding experience between mother and child. Disadvantages would be the mother's more prone to osteoporosis. It may be inconvenient, especially if the mom has to go back to work. There's inflammation and infections that can be caused, things like mastitis in the mammary glands. So just to name a few things. Now, Lactation is a positive feedback mechanism. So this means if nursing doesn't continue, the stimulus for prolactin release and milk production ends. Now you need to have both prolactin and oxytocin for breastfeeding, with prolactin contributing to milk production and oxytocin contributing to milk ejection. A letdown reflex is just this idea that sometimes oxytocin, this ejection of milk, can happen regularly when a woman is breastfeeding, but it can also happen when a woman hears a baby cry across the store, especially if it's her own baby, that oxytocin will get released and she'll start releasing milk or ejecting milk. One other advantage to breastfeeding, and it isn't 100%, but ovulation is often quite limited when prolactin levels are high. This means that when a woman is breastfeeding, she's a little less likely to get pregnant. And if we look at our last slide, look at that picture of that premature baby. Now, premature births account for about every 5 or 6 out of 100 births. But like I mentioned before, most babies born after at least 26 weeks of pregnancy have a very good chance of living with today's medical care. We have NICUs that have incredible pieces of equipment and nurses and doctors working in there to help treat premature babies and babies with health problems. A premature baby is one that has a low birth weight of 5 pounds or less. The average full-term baby in Colorado weighs around 7 pounds at birth. So like I've said throughout this entire lecture, premature babies often have medical complications. A baby born more than 2 months early is likely to have serious problems. One of the main problems in a premature baby is that lungs are likely to be underdeveloped as well as their liver. Jaundice often results because of liver problems. They may have other medical issues like low body fat because they didn't get those last months 8 and 9 when there's a lot of fat intake. They may have not all of their neural connections or myelination done yet, low body temperature, and low blood sugar levels. So that's it for development and fertilization. Make sure that you study anatomy and physiology. See everybody.